You're
Okay,
able to
so
see?
it's all yours.
Yeah. Thank you so much, Manu, and like so grateful for this opportunity. Though it is still sort of strongly ideated project, and I've been trying everything from my end to make things materialize because it involves a lot of scientific tools and the facilities to initiate and to perform these experiments, and it involves a lot of permissions and explanations as well. So, however, I mean, I could bring in some attention into this project. I'm working with a center called Center for Cellular and Molecular Platforms. It's a part of something called Bangalore Life Sciences Cluster. It's a sort of a non-profit organization, and it's a research organization come platform service provider. And there are lots of interesting programs that are funded by international and national agencies at CCAM. And I'm working with something called indigenization of molecular diagnostics. And here we try to help MSMEs and small and medium MSMEs and startups to or to bring in and develop their molecular diagnostic kits for different different diseases, different infectious and non-infectious diseases. Okay, so with Manu, I'm quite interested to develop a sort of antibiotic susceptibility test, especially for Staph aureus. So I'll just start with an interesting statistics that would tell you the gravity of the problem. Right, each hour of misdiagnosis or inappropriate antimicrobial therapy increases mortality rate by 5 to 10%. So we all know about antibiotic resistance and superbugs and everything, right? So there are multiple reasons, and if you talk about antimicrobial resistance, it's never ending. So there are lots of biological and non-biological reasons for the development of antibiotic resistance. Okay, so here I'm going to address only a very focused with a very focused approach. That is, how can we help clinicians to choose right spectrum of antibiotic so that they don't overuse or misuse broad spectrum antibiotics frequently. Okay, so we know like we do get infections, and there are people who are like quite vulnerable. They get infections, recurrent infections, and some are responding to certain antibiotic class, but over some time, after a period of time, they become resistant, right? So it's important for the clinicians to understand what is happening. So keeping that in mind, I thought like probably we'll focus only on Staph aureus because there's a wide variety of valuable research articles, publications available with a kind of scientific approach that I'm going to talk here. Okay, so the prototype has been named by Manu as Vesicle DX, where we are going to use membrane vesicles secreted by bacteria as a mode of diagnostics. Okay, so we do understand the priority is, in addition to developing molecular diagnostic kits for different bacterial and viral infections, it's important we also should be able to identify and test susceptibility patterns. Okay, on those lines, we wanted to develop a system that can identify antibiotics. Are you able to see the screen properly?
Yeah, we can see your screen.
Okay.
If you want, you can do presentation mode, but we can see it.
Yeah. Okay. So to develop a system that can identify antibiotic susceptibility pattern and resistance, especially for Staph aureus. So why Staph aureus? Because literature says, and a lot of studies are coming up which mention about something called extracellular vesicles or membrane vesicles, which are secreted by these gram-positive bacteria, and they provide a lot of survival advantage to this specific species. Okay, in terms of degrading the antibiotics they are supposed to be susceptible for. Okay, so and these membrane vesicles, not only they act as decoys, but they also help in horizontal transfer of mutant genes or genes that are important in antibiotic susceptibility. And in addition, they carry protein factors and other factors that would degrade or sequester antibiotics. So that is something which is very important and that we can utilize or use in application to understand its susceptibility to certain classes of antibiotics. Here it is beta-lactam antibiotics. Okay, so the organism is Staph aureus, and the reason is a lot of literally, I mean, was mentioned that they secrete MVs that are carrying these beta lactamases, which degrade the antibiotics. Hence, they provide survival advantage. And the samples that we can use may be urine and whole blood. And approaches, we need to isolate these membrane vesicles which are secreted by this antibiotic. So the principle behind the whole approach is when Staph aureus is exposed to a stress, it is going to release these or secrete these membrane vesicles. So considering antibiotic as one of the important stress factors, they immediately start secreting MVs as soon as it comes in contact with the antibiotic. Okay, so we can take multiple, we can take an array of a particular class of antibiotics, incubate these, Staph aureus in presence of these antibiotics, and try to see whether we can see the difference in the vesicular secretion pattern. If so, can we capture them and see and say like, or give a sort of a scale or a gradation based on the number of vesicles secrete, that how susceptible they are. Okay, so the plan was to isolate these membrane vesicles from the samples and do a sort of a relative counting and then lyse these vesicles, because it is well known that these vesicles secreted by Staph aureus carry beta lactamases, right? So if we are able to lyse them, you are exposing the enzymes to the antibiotics, then again you can measure the degradation of the antibiotic. So the prototype, in a sense, I could really think well in terms when it comes in terms of biology or designing biological assay. But to make it as a device, I thought like I was inspired by Manu's paperfuge and Foldscope approaches and tools. So I thought, why not we use paperfuge itself, because it goes almost to 125,000 RPM, right? Almost it reaches the centrifugal force that is very important to isolate these vesicles. So we can isolate these vesicles using a capillary-based method. And then if you're able to, bring in or identify the density space or the isolation space of these vesicles, then we can use light scattering properties to identify relative numbers. And then those can be then collected in a chamber where you have the lysis buffer. Basically, detergent-based lysis can help, and it exposes these beta lactamases enzyme. Right, and then we can have a sort of a fluidic arrangement where these enzymes are exposed to the substrate, and then allow the kinetic mode of reaction to occur, and then we can measure the OD. So, bacteria, Staph aureus, incubated with different antibiotics, releasing MVs. MVs are captured, relative numbers are identified, and then we are trying to estimate the degrading enzymes. Right, so we can have a sort of a correlation. So now I'll go maybe directly to the state part which I had made. Sorry, Manu, please don't get offended with the kind of a diagram I managed to make.
No, this is good. I think the idea is to make sure that we can communicate what we're trying to do. So this is all good.
Okay, so we have our paperfuge setup where we have these square capillaries, right? And we are able to manage to coat these capillaries with antibiotics of our interest. I've chosen this from a particular reference. It can be adapted or modified very well. But this paper specifically was very interesting to me because they had done how a resistant strain, which is this here, it is ST692, which you can see here. And you can see how the minimum inhibitory concentration has increased in leaps and bounds, right? So they are actually resistant to most of the beta lactams. So how these resistant bacteria, when exposed to these antibiotics, they secrete MVs and carry those beta lactamases, and those were put on the wild type, which is not mutated. And in a dose-dependent way, those MVs are able to allow the wild type to get resistant to the same beta-lactam antibiotic. So it was a wonderful paper demonstrating how the MVs play a major role in mediating antibiotic resistance. So I had chosen that as a sort of a preliminary antibiotic list here. Okay, so we can code them and allow the samples to be incubated there. And mostly references mentioned like between actually the secretion starts from 30 minutes and the max maybe up to 3 hours, so that you get enough load of vesicles to be, to be able to be detected at the end. And then you allow the incubation to happen. And then with a paperfuge, you are giving certain force so that the vesicles can be separated based on the buoyancy, right? And this is well known that vesicles have certain particular density so that they can be enriched in a fraction, okay? And it's important that we make an arrangement where this can be siphoned off into an horizontal tube or something, where a chamber-like situation can bring in the light scattering tool, and then we are able to catch the Brownian motion of these particles when they flow through the channel, right? And then, it can be measured. However, it cannot be absolute, as we know, because being a sample, whether a urine or a blood, there will be multiple other interfering particles, right? Even our own cell-secreted exosomes would be there. So it's important that we are going to measure the relative increase that is happening within that incubation period, right? And then after measuring the relative numbers, we are able to pull them, collect them in a place where you have your lysis buffer. Basically, tritonics or SDS-based buffers are more than enough to lyse these vesicles. And of course, wanted to mention a point that when we say membrane vesicles, mostly it's made of proteins, okay? Made of lipids. In case of gram-positive bacteria, it is sort of more towards proteolipids. So protein content is more because of the peptidoglycans that are rich in gram-positive. Okay, so accordingly, we'll be able to standardize all this. Only a few, combinations of detergents would help us to identify the right lysis buffer, and we are able to lyse them, right? And then it should be able to send to a separate channel where you have the substrate for the beta-lactamases, and allow them to interact, and on a kinetic mode, we'll be able to capture the substrate conversion. So that's what I've tried to capture here. Like post-spinning, we'll be able to distinctly see the vesicle enriched band. Or if we are not able to, then in case we are not able to get the band at the appropriate position with every time and with different samples, we can have an approach where you have multiple channels that can definitely capture different fractions. I know we'll be able to screen them based on the scattering property, whether that particular channel or band was having more vesicles or not, right? So then after the lysis, we are able to see the enzyme-based activity. So how I am going to take this forward, because I know this is scientifically heavy and it requires a lot of support and BSL2 facility to perform the whole panel of experiments, right? So I thought level 1 and 2 can be something which can be set up on the bench, and no, I don't need a really BSL2 facility to perform. So level 1 is basically to establish whether how many particle number can give a better scattering, and can we relate the particle number with the scattering property. Okay, so here I thought will choose liposomes between this size range 30 to 300 nanometers, which is what the membrane vesicle range is also expected to be. And then these liposomes can be, check whether the paperfuges are able to bring them in one at a one sedimentation density, and then we can take them for measurement for the counting using scattering method. So this would give us an idea whether these relative vesicle counts are obtainable, and can we match the number. Say, for example, I have taken 1 billion liposomes here, and what is the OD like? So we can have a sort of establish a control chart. And in the second level, where we can just look into the enzyme activity. Of course, lots of papers, they do talk about beta-lactamases based enzyme activity. So anyhow, we can try this as well, and then establish again a control chart where you have this conversion of 1 unit enzyme converting the substrate to this much, and what is the OD. So that again, we have to establish, which can be another simple experiment. And from third level onwards, we can bring in the cultures here. So I have mentioned both the wild type and the mutants here. I hope there won't be a much problem dealing with mutants. I'm trying to talk with Bugworks, an incubator here who's like doing wonderful job in discovering new classes of antibiotics for resistant bugs. Okay, so if we have the wild type and mutant, we'll be able to culture them, and we know the culture density. We can identify the ODs, and then we can harvest the conditioned medium where the secreted membrane vesicles are enriched, right? And take them and again repeat the same experiments where using paper fuge, I can identify whether it can be separated in a particular density. So all these can be done in non-coated capillaries where we are not bringing in any antibiotics here. We are just standardizing the protocol. And then again, do the cell counting and relate them. Okay, now we have started doing it with wild type and mutant. So we'll be able to see the difference. Are we able to capture those specificity and sensitivity that we are looking between wild type and mutant? And in level, I think from level 3, it must be done in BSL2 facility and taking care of appropriate disposal and everything. And level 4 would be like with the same culture setup as samples. We can again harvest the membrane vesicles, do the paper fuge based isolation, try to see whether the scattering can be this scattering is able to pick up the counts and number, and then couple it along with the enzyme activity. So this again would help us to understand whether all these can be integrated together, or do we find any challenges when we are trying to integrate different processes in the same setup, right? So again, this will help us to bring the control chart, like how can we relate the particular culture density with a particular particle number, and are we able to relate it with the appropriate enzyme activity, right? Then the level 5, what I thought is to bring in the antibiotics of interest. So we can have the coated ones, and then again with minimum inhibitory concentrations which are recommended, and then bring in these cultures, both wild type and mutant, and again repeat the same centrifugation, vesicle counts using light scattering method, and then lyse the vesicles and do the enzyme activity measurement. And of course, the final approach would be to use the sample and then check how it would work. So this is what I have thought of. I think level 1 and 2, I would be able to definitely do. I'm trying to speak with people, like, try to get some samples so that I can start with testing the first level 1 and 2 approaches. Yeah, this is what I have thought of, Manu. And in case I do, because this is a little time consuming, I know that very well, and know I need to really get appropriate permissions to perform. In case if I'm not able to take it further in near future, I have a plan B where it's much simpler, and of course I will come up with a proper plan for this, where this interesting chemotaxis property of bacteria can be again used to identify antibiotic susceptibility. I haven't come across any approach like this in any of the diagnostic methods. Here we know that bacteria has got they have their own ways of moving around in an environment. So whether there is an attractant or a repellent, the movements vary, right? The chemotaxic movements vary, and there's a lot of interesting signal transduction behind this. So this is a very simple method where when attractants are there, then they actually do the running motion, and as soon as they sense that something is wrong and something is repellent, something is toxic in the environment, they tumble and change the direction of movement. Right? It's a very simple phenomenon that we see, and this can help us to understand whether the bacteria is susceptible to the antibiotic, or it is sort of happy or not so resistant. Then it will continue its course towards running, right? So this is my another thought. Like, I think basically fluid computation is what is required here. I'm not very good in understanding this, but I think this can be a plan B if I'm not able to really take it further with vesicles.
Yeah, I think, Prabha, there are a couple of motility and electromotility types of assays that people have mapped, but not all UTI, for example, bacteria or other bacteria might have motility. So I guess
Yeah,
that's
yeah.
the
I thought
part
we
of
can
that.
think about certain class again,
Yeah.
equalize, for example. Yeah,
Equalize
it has to be. Yeah,
has to be motile. Yeah. Let's open to questions because I think we're going to run out of time. So on the phase 1 of the project, any questions, comments? Ray, go ahead.
Um, I just want to ask some like fundamental.
Yeah,
things,
yeah, yeah.
like how does the doctor know that it's Staph aureus in the first place? You know, does that like, and also how specific is this like vesicle secretion to this organism? Because from a doctor's perspective, honestly, if you are able to identify that it's Staph aureus, then you probably already have enough amount of the bacteria to do more traditional antibiotic susceptibility tests in the first place. That's one thing. And secondly, what clinicians usually do is that we will just give the strongest in the first place. And then after we know the antibiotic susceptibility, we will downgrade the antibiotic. So that's what people are actually doing.
Yeah.
So
Yeah. So Prabha, 2 specific questions, and I think this is a thread. So one of the general ideas of this is to really move drug-resistant assays outside traditional
culturing methods. Yeah.
settings. So, like the idea of going either culture, so the traditional time scale is between 6 to 12 hours. So Prabha, the first question is around ID. And I think the second thread, which is relevant on this front, would also be in terms of the vesicles. They are general to all common pathogens that have been identified, say, for example, in a UTI type of a context. And I'm assuming there would be another mechanism for the ID of the pathogen to begin with, which is not coupled to this as yet.
Yeah, right. So Staph aureus is something which I have mentioned specifically because compared to other species which also contribute for microvesicles, some also have beta-lactamases secreted in the MVs. Compared to that, Staph aureus is much more, I would say, strong or much more evident and prominent in terms of secreting MVs enriched in beta-lactamases. So if you see the ratio, because when it comes to sample, it can be multiple pathogens, right? It's just not only Staph aureus. But what is important is I have thought through this. For initial infections or the first preliminary screening, it may not be the appropriate method. But whenever there is a recurrent infections in a patient with a particular type, like doctors will have an idea like looking at the antibiotic history and the treatment, right? If it has been treated for Staph aureus or in general a particular class of antibiotics like beta-lactam, they'll be able to identify which would be the prominent or the maximum contributing infectious organism, right? That way, Staph aureus outscores other species which also contribute for MVs with beta-lactamases. So that way, it will help, but it will not definitely give any proper identification of the pathogen using this test, but at least it will give you whether the infection in that pool of microbes that are there contributing for the infection, are they resistant or are they susceptible to beta-lactam antibiotics or not? So that much of information is possible from this approach. And secondly, regarding the vesicles, Manu, what was the question specifically regarding the vesicles?
Yeah, I guess the question is, and lack of time, I think maybe one of the things we'll do a broader discussion, but just the general approach for this project is around the idea that Prabha has identified a large number of papers in the vesicle production, and we are just starting to build on that basic knowledge to say, can that be used in a diagnostic way? So it's not there completely, but one of the key factors would be is this is currently, this is much more of a transition from the fundamental science that yes, they generate these MVs. Now, can we extract any information out of it?
think in that aspect from the scientific part,
Mhm.
one very useful thing for the calculation is that to translate the units that clinicians are more familiar with to
Yeah.
how much vesicle that you can expect. Like, for example, the units that clinicians are familiar, it's usually like time to positive. That's what and so if you are able to know, like, for example, 12 hours of time to positive, how much bacteria does that correspond to, then how much vesicles you would be able to expect in that volume, that could help you to know whether or not the approach or the amount of samples that you are taking is that reasonable.
Yeah, I think probably we should definitely do a class of calculations in terms of, because we know the rate of MV production, we know for a given volume, the CFU unit for the sample, and that'll also sets challenges around, it's easier to spin smaller volumes at higher RPM than larger volumes. So that also kind of says that, oh, whether we actually need a pre-concentration step or not. But again, I think very similar to kind of what the discussion around the first team was, as you're putting together the Notion pages for the projects, it's very valuable to now where the design is to redo some of the numbers. So I think, yeah, we should do some of the numbers. And I'm looking